when you think about the halachic perspective, we have to add two more principles. And the first is the sanctity of human life, or covenant of human life. There are many examples. One that we discussed: can we use DNA technology to clone a Neanderthal human? Technically, we can do that. They have sequenced from all of the preserved, frozen Neanderthal tissue. We have sequenced the genome of the Neanderthal human life. There are 10 million differences between that sequence and all of us. There's technology available now, DNA technology. We can incorporate those sequences and take a human cell, make those changes, and create a cell that has a sequence very close to the Neanderthal human. And then we can take that cell, as you heard Eric so beautifully describe, using what's called iPS technology, to transform that cell into a stem cell and use that stem cell as a means of generating a fertilized egg, so to speak. We can implant that into a human surrogate, and 9 months down the line, we can clone a Neanderthal human. Are we allowed to do so? We would argue that that's not covenant agreement. Why? Because we're creating a species that is not as developed as we are. And for what purpose? What is the rights? In other words, similarly, we have the genetic capacity to take a couple, let's say they're both hearing impaired, and they want to have a child that's hearing impaired. Can we use PGD to select for that pre-implanted embryo that's hearing impaired? They say to the physician, we don't know how to raise a child that can hear. We can raise a child that is deaf. We know how to do that. We want a deaf child. Or two dwarfs who want to raise a child that has dwarfism. Are we allowed to do that? And probably the halachic response is no. We can't use these technologies to create defects. We can use these technologies to improve the health situation. So that's the first one. The second one, what Keller calls ethical relativity, or avoiding the slippery slope. Interesting, we all talk about the slippery slope. There are very few examples in halakha where a slippery slope has really gone into play. Rav Shabtai Rapport gave me one from the Gemara in Yuma, Pei Dalit, where the Gemara says, a Jew cannot tell a boy on Shabbos, save this patient. Why? Because maybe it'll lead people to think that only a goy can be Mechal Shabbat to save that patient. So on Shabbos you can't tell a goy to save the patient. You have to save the patient. Rav Hanani says not only that, but the biggest gadol door has to go out and be Mechal Shabbos to save the patient. That's one example of a slippery slope. In terms of bioethics, we discussed this. Can he use fertilizer? Can he unused fertilizer as discarded? And Rav Teller's position here, I think, is very unique. He says, to really discard the eggs is not appropriate. There's still a small, minimal amount of kedusha. What is the right approach? Donate those unused pre-embryos for research. That is an effective, ethical procedure that one can do. But the slippery slope in terms of secular bioethics is a problem. Because if you discard these eggs, will this lead to destruction of implanted embryo? So you have a difference between secular slippery slope and halakhic slippery slope. But these are the two additional guidelines that we are proposing. Now let me take you a little bit on some of the issues that we've discussed. We heard so beautifully what happened 10 years ago. 10 years ago you have a couple. They're both carriers for take sex. What were their options? The options were not to get married. And in fact, most Rabbanim 10 years ago, if a couple came to them and said, we're thinking of getting engaged, both carriers would say, do not find somebody else. Do not get married. The other option is to marry, not have children, which would die. The third option is to marry, screen the fetus using amniocentesis. And the fourth is to rely on Kodesh Baruch Hu, to rep time Hashem. All of these were viable 10 years ago. Let's go through some of them. Okay? Halacha says the following. In the Gemara and the Bamot, that basically don't marry into a family that has a genetic disease, especially as Rashi explains. And this has a really important warning. It says that the idea is you don't want to create families genetic diseases. But there's technology available to prevent these diseases from coming into the family. Use it at all costs. The other option, of course, is to adopt. This can a couple, lechatchila, get married, not have children, and just use adoption? Very difficult. A lot of postmen would say no. So those were two options. The third option is really what happens in reality. Even though we have Dori Sharun that is screened, Dori Sharun who screened many couples and told them they're not a match, if you actually speak to geneticists at Einstein at NYU, they'll tell you that even within the fruit community, right, they will get married, despite the knowledge, get pregnant, and terminate the marriage, terminate the pregnancy. And 10 years ago, how do you do amniocentesis? How do you do embryonic sampling? Usually happens at 11, 12 weeks gestation, right? It takes a couple of weeks to get the results. They have to talk about it. So usually termination was an option in 14 weeks. And as many of you know, there's really one, the Tzitz Eliezer was one of the few postscripts who would allow termination. Most other postscripts at that late stage would have not allowed termination of the pregnancy, even if they were carrying a Tay-Sachs or cystic fibrotic child. What about, well, I just discussed that. And one of the halachic problems is that simply most postscripts feel that only up to 40 days can you terminate the pregnancy. Then we go on to the last thing. What about, and again, in the Haredi community, there are many people. Shemer B'Kar Hashem, we say it all the time in Hallel. What does this really mean? And if you can look at the halachic analysis of this, read Rashi, this principle only applies when the danger with the sakana is very rare. It's a very small risk. And Chasam Sofer and others says that if the danger is known, if the couple knows the danger, then no one will be protected. You won't be protected. Shemer B'Kar Hashem only goes when it's a small risk and it's unknown. In the Ramban, Truman Tedeshen, says also if you're dealing with an educated individual who can ask, then Shemer B'Kar Hashem doesn't apply. So in our case, of a couple that know that they're both carriers of Tay-Sachs, and they have a 1 in 25, 1 in 4 chances of having an affected child that will die, the principle relying on Shabbat here, Shemer B'Kar Hashem, does not apply. Let's go to the test. What are new options available now both parties can do? Well, chorionic sample skills. The chorionic bill sampling can be done now in 30 days post-fertilization. There are two physicians, one in New York, New Jersey, and one in California, that have done this many times. Now, in chorionic bill sampling, what you do is you go in and take a piece of fetal tissue, not from the fetus itself, but the chorionic tissue. And it's not an easy or trivial procedure. There are a lot of risks. You need termination of the pregnancy, you need to birth defects, even if it's done in 11, 12, or 13 weeks. These physicians have developed a technology that they feel they can do it 30 days after conception. And, well, I don't have the statistics. There have been several babies that have been born, have been diagnosed healthy, and are born using this procedure. And what's the advantage here? Because in 30 days, right before the 40-day limit, as we all know, the Gemara says that before 40 days, a fetus is maya b'alma. It's considered like water. And it has a different halachic status than post-40 days. And many posts in America, Kettler and others would allow termination before 40 days of a pregnancy if the child was affected with one of these diseases such as Tay-Sachs or cystic fibrosis. So in these cases, they allow them to do this, but there is a medical risk. The other thing which we've heard a lot about is pre-implantation genetic diagnosis. And unfortunately, I don't have internet here, but I will show you a really nice movie. Let me see if I can get it on. If I do get it, you'll see how it really works. And let's see what. No. No, it doesn't work. I'm sorry, I apologize. But anyways, let me give you a brief synopsis because it's really complicated in one sense. You take the egg and the sperm, you fertilize them in the test tube, you let the cells, as you heard, divide until 48 hours, and you remove one of the cells. It's a miracle that you can remove one of 8 cells of pre-implanted fetus and it has no effect on the fetus. It's really unbelievable. And you take that one cell, that's the other thing, that our technology is so good that you can take one cell and do complete genetic analysis on it. And you can test for various diseases, and what you do, as you've heard about, is that you have to isolate 10 to 20 eggs, fertilize all of them, and for Tay-Sachs, until 25 are going to be completely without any dependent genes. And you have a choice of implanting one or 2 of those. And today the technology is so good, usually you'll implant 2, 1 will take, and there's about a 30 to 40% success rate, especially in young women, in getting a healthy child. What can you test for? I just give you a few of the diseases which really the correlation is very good. Okay? Non-diseases, gender. What's interesting about PGD is there is only one example in halacha that I know of in which you can use PGD to select for gender. And that's the following case. The case is of a Kohen, correct? A Kohen who is infertile. Now, what does he want to do? He wants to use a donor sperm to impregnate his wife, which you're allowed, many Muslims allow that. And usually if it's a donor sperm, it should be a non-Jew. We'll discuss that in a little more detail later on. And the issue is, of course, very profound. And the issue is that now this child is born, it's genetically not related to the father, it's the Kohen. When the child goes up for an aliyah, it's not going to get a Kohen. The whole congregation, the whole community is going to know that this child is not the child of the Kohen. It's a busha. So under those circumstances, many posting will allow to do selection for gender. So you can select a female child, a baby. So this way, the family will not be divided, and the whole community doesn't have to know where the child came from. That's the only example that I know of. Now, when you think about these procedures, you have to consider the medical risks. Remember, you're taking a woman who's going to undergo PGD. You have to hormonally hyperstimulate. The iris is associated with that. You have infections, pain, syndrome called ovarian hyperstimulation syndrome, which could be blood clot, kidney failure, shock, edema. There are real risks. It's not just good. We're getting better at this, and you'll see in the future what's going to happen. Even with the Bar-Ilan sampling, the iris would use a determination pregnancy. So whenever you make these calculations in terms of halacha, you've got to take into consideration the medical risks. The other halacha, what are the halacha considerations? The first one is if you're going to engage in PGD, that couple has to undergo contraception their whole life. Okay? And what contraception do they use? Are they comfortable with this? Contraception is not a trivial halacha issue. What contraception should they use? If they decide to do termination of pregnancy before 40 days by using CVS, at 30 days by these new technology, then they can have very normal relations, no contraception, and there are other risks involved. And I thought I would show you what the difference is between various embryos, because people don't appreciate this. Here's a 28-day-old human embryo. This is a 40-day-old human embryo. You can almost tell. It looks a little bit human. This is a chemical. So what is the significance of 40 days? There are different theories. We don't really know. That's on the soda. But it certainly has some strength. And I think it's important. The third thing I want to mention is what happens to the child. When you do PGD, timing of these hormones is so critical. When you give the first hormone, when you extract the eggs, when you fertilize, when you implant. And it's over the course of many weeks. What happens if it's over the Chagim? Do this on Shabbat? Can you do this on Yom Tov? How do you do this? In America, it's not such a problem. Why you're lodging to these procedures? You're allowed to do them Yom Tov because in Germany you have a board. You have a non-Jew engaged in these procedures, and you're allowed to do this. In Israel, I really don't know what they do, but it is more of a problem in terms of how do you overcome the prohibitions of the Shabbat to do these procedures. There are solutions, I'm sure, but just throwing this out. Now, what about the future? Here, I think we have something really important. And it's a message that only addresses to the Orthodox Jewish community. There are new technologies that are being developed, not necessarily for this type of intervention or diagnosis, but can be applied. And that is to obtain fetal cells from the blood of a pregnant woman. Because as we speak now practically, as Eric will tell you, if we have 3 fetal cells, that's sufficient to do genetic testing. We're almost at the stage where 1 fetal cell would be sufficient. And in a pregnant woman, you have fetal cells escaping from the fetus and circulating in the mother's blood. So we can have, and I can tell you different technologies that we can implement to obtain a cell and isolate a fetal cell from the blood of the pregnant woman before 40 days. She doesn't have to go PGD, no hormones, get pregnant normally, and isolate that cell. And 95% chance of success of diagnosing whether that child is affected with Tay-Sachs or fibrosis. And at Columbia, they're doing obtaining fetal cells from vaginal secretions in the first 3 weeks after conception. The same procedure. They collect the vaginal secretions, there are a lot of fetal cells, and they can use this. And the advantage is you have fewer medical risks to the mother. And you see we're approaching a state where we're not necessarily going to have to use PG. If we can use and obtain fetal cells or fetal DNA, that's another area. And what's interesting that I can tell you is that the push for that is not as great. You would think that if we want our community, we should be investing $1 million to get this technology implemented within a couple of years. And the only group that should invest in this technology and develop it, because there's a real need for it, and it creates a lot of solutions to problems that you've all heard about this morning. And I would push that if somehow we could find a divorce, to give the $1 million, we could get this going within 2 years. It would be a sure. Okay. What about other? And this is really the gray zone, as everyone said. What about using DNA or stem cell technology in other areas of reproductive medicine? Let's look at the first one. Use of DNA technology to screen for growth. As you know, as I told you before, if a couple is infertile and the man wants to use a donor sperm, most postscript would say we use donor sperm from a non-Jew. Why? Because of the minute suffix that if you use donor sperm from a Jew, that son or the daughter from that artificial reproductive procedure might marry a karov, a relative, and that's one of the arayot of the Torah. And because that small suffix, and as Rav Shabtai Rebbe said, it's really minute probability. Because of that, we say do not use Jewish donor sperm. Now we have new technology. You have Avram and Sarah. They are infertile. Avram decides he wants to use a Jewish donor. He wants to use a donor from David, Jew. Now Yitzchak is born. Yitzchak grows up, wants to marry Rivka. They go to Dor Yeshurim, and they sign a sheet that says, Yitzchak was born by artificial reproductive technology. So now they can use DNA technology to establish whether Rivka and Yitzchak are Krovim or not. How would halacha deal with the use of that technology? How much confidence or statistical accuracy would be required to remove the suffix? Another thing is related to surrogate mothers. I know there's a push here by PUA to say that if you have a surrogate, that is a woman, you have a couple that the woman cannot carry a child. So they hire a surrogate, And the question is, who's the halachic mother of the child? Is it the surrogate, or is it the biological or genetic mother? And Rav Shlomo Zalman Auerbach, years ago, said, you know, this is one of the few cases in halacha there's no halachic precedent for it. Therefore, we go both the surrogate status and the genetic biological mother's status. And that's lechumra. And that's how we pass it. Now, who wants to say, let's go genetically? And I think, personally, this is a wrong attitude. So, we discussed this. We can skip that. This is the certainty element. This is something that Shakai really spoke about. But I just want to say that genetic results, both, as Jack mentioned, both Shlomo Zalman and Waldman ruled that you can use genetic results as a consideration in final determination. That's just an added thing. But let's go back to the surrogate mother. Today we know that being a surrogate is not an artificial process. What we know established is that fetal cells escape from the fetus, implant in the mother. We're talking about millions of cells that implant in the mother and stay within the mother for the lifetime of the mother. They implant in the brain, they implant in muscle, and what function do they have? We don't know. I mean, a woman who had 5 boys, does she now want to play basketball? I don't know that. But there are effects these cells. Listen, you heard about junk DNA, right? If you look at the whole DNA genome, only 5% is used for encoding genes, and the other 95% used to be called junk. But we know Kadosh Baruch Hu does not create junk. That's a rule we can believe. And same thing here. If these cells are implanted in the mother, there's a purpose for these cells. What it is, I don't know. But there has to be a purpose. And it's even more profound because normally, when a woman is pregnant, right? When a woman is pregnant, her immune system is, in a sense, compromised to allow her to carry this forward in the body. This is not our own cells. Part of it are our own cells. And now, after she's pregnant, the immune system is geared up again. And why doesn't she reject those implanted cells? And how do we know that she has them? Because you can do an autopsy on an 80-year-old woman who's had boys, and in the brain, the muscle, you see cells that have both the X and the Y chromosome. And the only way she can get those cells is by getting them from the fetus. It just doesn't, because she's XX. So we know they're there. But not only that, in the last 6 months, we know that not only do cells go from the fetus to the mother, but stem cells from the mother implant into the fetus. What are they doing there? We don't know how many. We don't know what effect it has. So the third consideration is that the impact of pregnancy affects the personality of the child. Any mother who's carried a child will tell you, oh, this child I was in this bad mood, that's why this one came out like this. As Rabbi Weinberg said, the environment affects the personality of the child. That's clear. So a surrogate is not just an equal. A surrogate impacts both on a cellular level and on a physiological level. And we know from halacha, why is a Rav considered kavod avayim? Because it affects the personality of the person. So I would argue that in terms of surrogate, we have to go back to Shlomo Zalman, that both the surrogate and the biological mother should be the halachic mother. And there is a remnant in the Torah. It's a little cute. Moshe, in Balot a few parshas ago, he was complaining, right? Oh my God, what are the people yelling at me for? And he says, Ha'amuchi, Hariti et kol ha'am hazeh, im anochi yiladatihu. And the Rambam says, calling on the mother. Notice the language. Did I conceive this nation? Was it I who gave birth to them? He could have just said, did I conceive this nation? He could have just said, did I give birth to them? Why does he say both? I would offer to you, but both are equal. Only asma. Okay, so now we're left with some really interesting ideas that we don't have any conclusions, and I think this is what we have to think about. First of all, using stem cell technology, we can generate sperm-like cells from skin cells. Now, this is very profound. I can take, it's not real cloning, but the closest we can. I can take well, we do this now real. We can take a couple that, let's say, the man did not have sperm. We can take his skin cells. We can transform them from a 46 chromosome cell to a 23 chromosome cell by a meiosis process, and we can use that cell to fertilize his wife's oocyte, and they have healthy children. It's been done. Several babies have been born this way. And with that technology, we can do a lot of other things. We can take a woman, take her skin cells, transform it into 23 chromosome gamma-like cell, fuse it with her own egg, and create a very close clone of that woman. So can we clone human beings? The technology is there. It's not an exact genetic clone, and if you want to hear more about that, I'll tell you why there's epigenetics, but it's pretty close. So you can generate sperm-like cells with skin cells. Not only that, but we can use stem cell technology to create very interesting animals. We've used stem cell technology back in, this is an interesting story. Mike McEwen was a graduate student with me at Rockefeller back in many years ago. And he went to get his PhD, MD, he went and did an internship in infectious diseases, and he went to the lab of Irving Weissman out in Stanford. At that time, in the early 80s, late 70s, Irving Weissman had a collection of mice, unbelievable mice, of different disease models. This mouse looked like this disease and that. And he went to Mike McEwen and said, Mike, you pick whatever mouse model you want and study that, and that will be your postdoc. He went and did a clinical analysis and found this one mouse called the skid mouse. It had no immune system, had no immune cells. He said, wow, this looks to me like AIDS. Looks like HIV, and that had nobody knew anything about HIV. It was a really new disease. And he says, you know what I'm going to do? I'm going to take bone marrow, human bone marrow cells, and I'm going to transplant it into this mouse, and he created a mouse with a human immune system. And he created the first model to study HIV in mice. And a lot of people have been using this model. We understood how the virus replicated. We got drugs from that. So taking and reconstituting the immune system in a mouse, a human immune system, was done back in the 80s. Now they've done this with sheep. We can create a sheep. You have an individual, let's say, who needs a liver transplant. Very hard. You know what the list is to get a liver transplant. It's very difficult. We can take his bone marrow cells now and transplant them. We transform them a little bit into precursors for liver and transplant them into the fetus of a sheep. Now that sheep born 6 months down the line will have a liver that's 85 to 90% human cells, completely compatible to the person who needs the liver transplant. And we haven't done this yet, but on a theoretical level, once you're over 90% of immunocompatibility, you can do a liver transplant and you'll be fine. There may be other problems, but let's not discuss it. So you can create animals with human organs. Is there anything ethically wrong with that? Absolutely not. But what about creating a cow with human beings? We can take, and theoretically hasn't been done, and create a human university cow. We have a wonderful incubator now. Take a embryo, put it into this cow. What is it? Is it human? It was one from a cow. Is it a cow? And not only that, and you can say, is this any better than what we do now? Now a baby's born, right? Prematurely, where do you put it? Put it in this black box. Is a black box any better than a cow? But there is not, we all know, because Lechatchila initially were trying to create this baby by natural means. It got sick, and we have no choice. For that baby to survive, we have to put it in this black box. But here, to use a cow, which is an artificial incubator, may create what we call a stigma against covenant abuse. And then we can do the following. We can engineer mice. And this has been done. It's been published. There are mice that produce human sperm and human eggs. So a woman, she's 20 years old, and she says, you know what, I want to get my PhD, my law degree. I'm not going to have time to have children. But I know when I'm over 35. it's going to be a problem. Might start giving birth then. The incidence of Down syndrome is higher. What do? I'll have a colony of mice in my basement that have my eggs. Right? When I'm 35, my uterus is fine. That's not the problem. I come with my eggs. So the uterus is fine. I have these mice. I isolate. They're reproducing all the time. Fresh, beautiful oocytes. And well, we haven't really fertilized it to see if it works. We have that possibility. So these are things that you're going to read about. So these are some of the challenges we have to think about. We discussed that couples who are both carriers of monogenetic disease marriage. Should we use the new technology? We have an obligation to develop that knowledge. And I think we do. Can we now change our directive? Wouldn't it be nice if halakha would say, you know what? You can use a donor sperm or donor egg that is from a Jew. Because it makes life a lot easier. We all feel that there's something special about having our own culture in our offspring. And how will halakha deal with future situations, such as an animal that's engineered with a human uterus or a human brain cells? I didn't discuss that. One of the things that was a real bottleneck was a few years ago with Irving Weissman. He said, you know what? I want to study the brain. So you reconstitute 2% of a mouse brain with human cells. At what point will we be able to reconstitute a primate with human cells, and what will that do? And this is not pie in the sky. Today, as we speak, there's a very interesting gene called FOX2P. FOX2P. Okay? And 2P. Right. It is a gene that encodes for language. There are families that have mutations in this gene and they can't speak. Now, what's wonderful about this gene, its difference between its form and the form in the chimpanzee or the non-human primate is only 2 amino acids. 2 amino acids differentiate the human form from any other species. And what did they do a few months ago? They took the human gene and they transplanted it into a mouse. They weren't that courageous to do it into a primate, a non-human primate, chimpanzee, but into a mouse. And they looked at the development. Now, of course, the mouse did not know talking, but its vocal changes were there. But more important, you saw changes in the cerebrum and the parts of the brain which were remarkable we never saw. So this little gene has profound implications in human development and neurological development. Could we could. There's no problem scientifically taking this gene and putting it into a chimpanzee. If the chimpanzee would apply to my phone, would you accept it? If it doesn't, you have to change his phone. Probably, but it could maybe understand at what point, as raise this, at what point does halakha consider a person being a human? What?
He loves chamor.
But these are real situations that we are going to face and we have to deal with. And finally, as Rav Shabtai Repo really brilliantly said, what percent accuracy will DNA technology have to meet in order to impact halakha standards of search? So these are the type of things that we're going to face. This afternoon we'll discuss gene patenting, gene privacy. And the other issue which we haven't discussed, which is a whole symposium itself, is that who does this information belong to? If you are diagnosed with a genetic disease, who do you tell? Do you tell your sister, your relatives? Do you tell your children? When do you tell your children? When do you tell your future wife or spouse? When on the first day, hey, my name is John, what's your name? I'm a carrier of Pesach. Think about it. At what point to do these? These are really practical implications. And we have to analyze it. So what I've done today is try to give you some of the futuristic applications of all this technology, and which we really need some input from the halakhic authorities to find out. The Rebbe told me before I left, he said, this is the message I should give to the Kehillah. We say every day, there's a Musa hospital. In other words, it says, every day God is creative, every day it is creative ability. So what's the Musa? Why do we say this? We know God is creative. The Musa is that we are partners in these creative process. And just like God is creative, we have an obligation to use our creativity to help mankind as well. And I'd just like to acknowledge Moshe Tyler and
Scott.